Thank you very much, and I'm absolutely delighted to be one of the speakers in the dkNET series of talks. What I'm going to be describing is the collaborative Microbial Metabolite Center, and really the collaborative way that we're capturing community knowledge associated with microbial metabolites. But before I highlight this, I want to showcase the companies and that I co-founded and that I'm an advisor for to make sure that those disclosures get across. So why do we care about microbial metabolites? I think if you really think back in 2007, when the initial human microbiome project was launched, we didn't really, we knew a little bit about microbial metabolites and how they play a role in human health. But really, with that first human microbiome project, we all of a sudden realized just how many different microbes existed and lived in and on our bodies. And since then, we've started to learn how these microbes contribute to human health. Now, one of the major ways that they contribute to human health is through the metabolites that they produce. And oftentimes, this is really a yin and yang. Just to show you two examples of the type of molecules where you can sort of think about the yin and yang of microbial metabolites, because in some cases they're beneficial. So ILA, for example, can reduce inflammation, whereas colibactin tends to increase inflammation, and it tends to be associated with inflammatory bowel disease and colorectal cancer. In fact, it's a DNA intercalator, and it actually cleaves DNA. And so you can see how that can promote cancer. But one of the challenges when studying microbiome-derived metabolites, and you really want to start to begin to think about mechanistically interpret the microbiome and the functional role of the microbiome, and in relationship to all the metabolites that are produced, is that there's not really a good resource to go to find out what the functional role of these metabolites are, even what are all the microbiome-derived metabolites that exist. And so my laboratory, I do a lot of untargeted metabolomics data. And the way to think about untargeted metabolomics data, you want to see as many molecules as possible. It's very similar to if you do a metatranscriptomic experiment or a metagenomic experiment. You want to get as much sequencing information as possible, and then you do data analysis with it. And so one of the things that you do with untargeted metabolomics, or many people will do, is you try to annotate the information that's embedded in it. Now, when we did this in 2016, we had an average annotation rate of 1.8%. And so 98% of the data remained uncharacterized at that time. This has now grown on average to 14% of the data is annotated. But now let's look at these metabolites and ask the question, okay, for the ones that can be annotated, only about 1 to 3% of those 14% we can actually assign to be of microbial origin. And so this is in sharp contrast to when, let's say, you take a fecal sample and you subject these to metagenomic sequencing. What you'll see is that the majority of genes that you can annotate are of microbial origin. And so there's a real disconnect between what we observe based on sequencing versus what we can annotate in terms of metabolites that are produced, especially since we know that microbes are powerhouses of metabolism. So you would expect them to be able to produce lots of different metabolites, but those are not well characterized, or most of those are not well characterized, I should say. Now, we're not the only ones that recognize this. NIH put this wonderful call out where they wanted to create a microbial metabolite knowledge base. And it was really a contribution of 6 different centers at the NIH. And it's clearly an indication that all of these different centers and programs recognize the value of microbial metabolites. And they also recognize that it's really tough to mechanistically interpret the microbiome because we don't have a good place to really look into these microbial metabolites. So what we proposed was to build a microbial data portal upload system, a knowledge capture system with searches and analysis capabilities. And this is done with Ming Wang, who is now an independent investigator at UC Riverside, and myself. And we basically enable the data to be reused and that it's ready for analysis. And this we also do together with the Rob Knight, who's also a true microbiome expert. But he also has a lot of experiences making data available and reuse, and really in the context of how a microbiome investigator might use this information. And then in order to be able to use this infrastructure, there needs to be documentation created, there needs to be training provided, which is Ming and myself are responsible for that. And we'll be organizing a yearly CMMC meeting, although the past year we replaced this with hands-on workshops, so that we can train people in how to use this particular ecosystem. And so the idea is that together with Bandera, who's an expert in database curation and really knowledge enrichment strategies, that any data that comes in, that the knowledge gets enriched, and I'll show you how we do that. Now, where does the microbial data come from? There are accompanying R01s that have been awarded that are discovering microbial metabolites and their activities, and that all that knowledge and data is supposed to go into the central repository. Now, we've expanded that to anybody who is interested in microbial metabolites worldwide, because in the end, the goal is to try to improve our ability to mechanistically understand the microbiome. So the more data we get into it, the better that will become. And also in terms of knowledge enrichment, we're starting to interact with other resources that exist to enhance the enrichment and the information that we can get of these microbial metabolites. And so the first focus that we spent some time on that we needed to create was a microbial metabolite knowledge upload portal. And basically it's there to capture structural knowledge, sources of molecules, as well as phenotypic activity information. This is then connected to a tandem mass spectrum. So if you fragment a molecule, you can break it apart. It's called a tandem mass spectrum. And you can think of this as a unique barcode, as you will, to which we now connect all this knowledge, basically. Now, these barcodes are linked throughout the world with universal spectrum identifiers. So this could be in our own ecosystem, and I'll highlight the ecosystem in a second that we have, but it could be in metabolomic workbench, it could be in metabolites, it could be in PRIDE, wherever the data is stored, we can create a universal spectrum identifier, and it reports on the project, the files that it's in, as well as the individual spectra that are associated with it, so that now we have a hard data linkage. And so these USI are also captured to a larger knowledge base, and all of this is indexed. And then the goal is to enrich the information of what is stored, so that we can get observations in what organisms where it's been observed in, maybe health association, intervention associations, so phenotypic associations, what kind of tissues, biofluids it's been seen in. So these are the kind of things that we try to use to enrich. And really what we leverage is public metabolomics data to enrich this information. And it's really all possible because we index all that information within our GNPS ecosystem. This is a community, it's a community infrastructure, but the people that are responsible for building GNPS is really shown here. Myself, Nuno Bandera, and Ming Wang. He started this as a graduate student and continues this to date in his independent career at UC Riverside, and certainly a key contributor to the work that I have shown here. Now, what is GNPS? Well, it stands for Global Natural Product Social Molecular Networking, and it's really a knowledge capture and analysis ecosystem that is built with and for the community. And I think that's really important. It's with and for the community. So it's not just us that is building this. No, it's with and for the community. And so what does that look like? The initial infrastructure that we build is we have a data repository, and this is built on top of the massive repository supported by NIH for the proteomic storage, but we have a component that also collects metabolomics data, particularly untargeted metabolomics data. We also capture knowledge, so any annotation that you have, any known molecule where you have spectra for, or if you have metadata that's attached to it, we try to capture that into this ecosystem as well. And so this has now grown to 3,400 projects, about 800,000 LCM-SMS files. And if you have to think about it, let's say the average cost of an LCM-SMS run, just to run itself, not to the cost to get the samples themselves, is already $80 a sample. So you can see how, the enormous resource that this is creating and the costs associated with it to be able to get this data. But because one of the things that we did within this particular ecosystem is we harmonized all this data, because there's about 30, 40 different vendor formats of data in mass spectrometry. And so we decided, if you want to make linkages at the data level between one data set and another data set and learn from each other, you need to have it into a format so that you can build these connections. And so this afforded us to build, at this point, the community really has built 50 different analysis tools. But also, once you do analysis inside the GNPS ecosystem, it is now data science ready. And so now you can make the data outputs ready for third-party tools to do statistical analysis in MetaboAnalyst. You can do visualization in Cytoscape, in Chime, do high-level statistical analysis. And there are lots of other tools, the third-party tools that the output is compatible with. And so, but we've spent a lot of time in the last year and a half really on this indexing and linking to the universal spectrum identifying. And this is just an example of a prototype what indexing truly enables, and that is really fast searches. That's one thing. It enables lots of other things, and you'll see a couple of examples. But fast searches was important because we started to have to do searches at 1.2 billion MSMS spectra. And there was no ecosystem that was capable of doing this. And so the indexing really facilitated this. And so we actually just in the past 2 days, I spent time with Claire O'Donovan from Metabolites and Subramanian from Metabolomic Workbench. And we're working with them to also index their ecosystems and link them to USIs. And what that means is that soon we'll be able to search all 3 repositories pretty much simultaneously. So the 1.2 billion will increase very significantly. But now you have this system in a uniform format. And so now you can build search engines. So the first search engine that we built was MASST. It may sound familiar, it sounds like BLAST, and that's because it actually serves a very similar purpose. When you go to NCBI, you do a BLAST. In BLAST, you can paste a protein sequence, an RNA sequence, or DNA sequence, and you can find all the data sets, all the other data sets that have been deposited in the public domain that have that same sequence or analogs of it. In other words, mutation splice forms. So you do based on the sequence alignment. Well, we now can do the same thing for molecules, where we could search our fragmentation spectra for in each molecule that is detected, and we could search if that same fragmentation spectra has been seen in any of the public data, or if there are any analogs, so related molecules that can be found. And then we have a MassQL tool that allows data pattern filtering, and I will discuss that later in the presentation. But having this ecosystem, I mean, the Collaborative Metabolite Center really leverages this entire ecosystem to make data reusable and also to be able to enrich the knowledge that's associated with any microbial metabolite that gets entered into the CMMC knowledge portal. And so the first thing that I want to show you is the creation of the Microbial Metabolite knowledge portal. And this is what the website looks like. It's a pretty straightforward. You can do single upload or batch uploads, and there are specific templates available for this. But the key thing that we need is a universal spectrum identifier. So this could be of an unknown molecule or a known molecule. As long as you know that it's associated and made by microbes, it can be entered here. That's the important part. If you're interested in adding to the knowledge base and you don't have an MSMS spectrum, reach out to us. We're happy to collect MSMS spectra on the compound of interest. This upload portal was created in one-on-one meetings with individual R01 grantees, and we do expect that it's going to evolve as needs arise with the community. So as we said, the universal spectrum identifier for a given molecule is required. If you have structural information, you can add that information. You will specify the origin of the molecule, whether it's microbial metabolism of drugs, microbial metabolism of food, human-made molecules, etc. Then we try to link to taxonomy or phylogeny. If you have an NCBI ID, you put the NCBI ID there. But in many cases, you only have sequencing information, and you provide that the sequencing links of where the sequencing is stored, so that people can map it taxonomically and phylogenetically. If you have biosynthetic information, or in other words, the genes that are responsible for making it, that'd be great if that is in MIBig. And so MIBig is a resource that connects structures to the genes that make small molecules, and many of these are microbial. And so if there's an MIBig session, all the information is stored. If this is new information, you can add and enter it here, or you can enter it at MIBig. We also specify the source of the metabolite. So there's a lot of microbial molecules or molecules that are made by microbes that are also made by other sources, such as the host, or that may come from diet. And so you would indicate that. So serotonin is an example that is found to be produced by microbes as well as the host. GABA is another example. Glycolic acid is nowadays also an example because it's been shown to be made by microbes as well. Then there are activities that are documented, and this is a bit free form at this point because there's no systematic activities that are being reported. For the ones that have systematic activities, they will be consistent throughout. And then we try to record grant information, of course, any comment that you have, references. Grant information is important because that will show to NIH how many different grant sources are benefiting from this particular resource. Now, all of this information goes into this knowledge base. So it's a CMMC knowledge base. And we have both a USI-centric knowledge base and structural entry-based knowledge base. How can you reuse the information that is captured? Well, the simplest one, if you want to download it, you can download it, and you can do whatever you wish with this particular resource. We don't feel that we own it, but the idea is that everybody who is interested in microbial metabolism, microbiome-derived metabolism, and their functions, that they benefit from this particular resource. The second way that you can use it is through enrichment in molecular networking. And I know many of you might not know what molecular networking is, but when you use the GNPS ecosystem for analysis, and I will tell you in a little bit what molecular networking is, but the important part is that when you use GNPS to do analysis, it creates like a blockchain. Basically, it's a permanent link to the data analysis, so it's easy to reproduce. You can clone it and then do the same analysis or slightly variations of these analysis. And that blockchain can now be imported into the CMMC search, and it will search the knowledge base so that now you can import it into Cytoscape and really visualize which ones are coming from microbes, what microbes are able to produce it. And so to just show you how this works is you first need to understand how molecular networking works. So if you have a sample with a complex mixture of metabolites, in untargeted metabolomics, we fragment all the ions that are observed. So we get these fragmentation patterns. We then merge all identical spectra and represent those as nodes. And each of these nodes, the way you can think about is they represent a unique molecule. It's not a perfect picture, but for the sake of this talk, that's how you should think about it. Each node is a molecule. Then we can do spectral alignment, similar to sequence alignment, where we can look for related molecules. And then you can create a summary. Basically create a network of all the molecules that are detected in your sample or your project or whatever analysis you're doing. And so when we take this network and we now merge it with the CMMC knowledge base, we can begin to visualize, for example, here's one of the pieces of metadata that's captured in the knowledge base, if it's microbial or microbial and diet derived. So here we have an example of bile acid conjugates that are all known to be microbial derived. And then here we have gamma gluteoglycine. We know that this is both diet derived, but also that microbes are able to make it. And so that allows you to give another level of interpretation. But notice how many nodes are still not colored. And that's because the reference database right now, although it's pretty good, 6,000 MSMS spectra are curated, it's just a small drop in the bucket. And this is really where we need to bring the community together and say, hey, can you help curate it? And so we've started to make these calls to begin curating this knowledge base with the larger scientific community. We have office hours for people who want to get ask questions. We meet one-on-one with people, but both the grantees, but as well as the expanded scientific community that is interested in microbial metabolites can contribute to this, and we welcome contributions to this. So the third mechanism to leverage this data in CMMC is really the creation of the enrichment strategies such as MicrobeMASST. Now, the idea is that if you have an MSMS spectra, even if there is no structure known yet, that you can learn which microbial taxa are able to produce these particular molecules. And so to build MicrobeMASST, what we did is we leveraged the GNPS and redo ecosystem. So you heard about GNPS. Redo is basically a strategy to control the metadata associated with each of the files that go into GNPS. And this controlled metadata is super important. What I did is I reached out via social media. And within a few months, the final paper had more than 100 scientists that participated in this. And we curated 100 million MSMS spectra and connected them to their bacterial, fungal, and human cells that are able to produce them. And the reason why we added human cells as well, so that you can begin to start thinking about differentiating what might be a human-derived metabolite or host-derived metabolite versus microbial metabolite. So if we look at the taxonomic tree itself, the unique taxonomic names that were provided on the 60,000 files were 2,300. Those were then blasted. That gave 1,800 unique lineages. So the final taxonomic tree had 2,800 nodes. And here's sort of the taxonomic distribution associated with this. And so now when we combine this with a fast search, and this is really important because searching 100 million spectra wasn't a trivial thing to do. But in the last year, we've solved that problem by indexing. And so now we can have a MicrobeMASST where you can search, and within seconds, you will get a taxonomic tree like this when you search with an MSMS spectrum. So in this case, I'm showing the MSMS spectra that was search of commendamide, which was a microbial molecule that was discovered to assist in lowering sugar levels. And there was one microbe that had been shown to produce it. Here we actually show that many microbes, especially microbiome-derived organisms, are able to produce this particular molecule. But what I really like about MicrobeMASST is that you actually don't necessarily need to have a structure attached to the MSMS spectra. So let's say during your studies, you discover a candidate microbial molecule, and now you want to verify if this is the case. Well, MicrobeMASST is now a really good strategy to look at this. Now the paper, this is really the first successful paper of the CMMC. The paper has now been published And we're just absolutely excited. We've been so excited for on the feedback on this particular tool by the community, particularly the microbiome field. So what I want to do next is show you how we can use some of these tools within the CMMC to discover and really enrich what we know about new metabolites. And the focus here is just on microbial bile acids. And that's in part because we discovered some really interesting microbial metabolites a few years back. Now, some of you may not look at bile acid biology routinely, so I'm going to give you a little bit of background behind it. So bile acids are made from cholesterol, largely dietary cholesterol. And so if you have cholesterol, it gets converted to cholic acids or chenodeoxycholic acid in the liver. Then they're conjugated or really amidated with taurine and glycine. And then those are stored in the gallbladder. And what's remarkable is that while they sit in the gallbladder, they are they're possibly the most concentrated molecules that exist in the human body. They've been observed in concentrations as high as 300 millimole, which is just extraordinary. I still can't wrap my head around it. And you heard that right, 300 millimole. But as soon as you eat, the gallbladder empties into the duodenum, down the digestive tract, ileum, jejunum, all the way to the lower digestive tract. And when it enters the digestive tract, microbes start to act on it. They start to remove these conjugations, but also they change the decorations of the hydroxylation states on these bile acid cores. But what we found in 2020 is that microbes were not only able to hydrolyze these, but also actually conjugate new amines to it. And in this case, we showed phenylalanine, tyrosine, and leucine. And so the question that we had is, okay, if there are 3, are there more? And how many more bile acid modifications can occur, and how do we find them? And this is where MassQL comes in. So MassQL is really, you can think of this as a pattern-based filtering mechanism. And it really, all of mass spectrometry, irrespective if you do imaging mass spectrometry, glycomics, proteomics, lipidomics, metabolomics, doesn't matter what mass spec you do, all of mass spectrometry data is defined by only 5 characteristics. And so we should be able to come up with a language to be able to filter this information. And so the language itself is precursor ions, so the MS1, so the intact molecules and their isotopes. If you break up the molecule and you fragment the molecules, you get the fragment ions that you can look for, or the delta masses. If you do chromatography, you have retention time information. If you do ion mobility, you have drift time information. So this is every mass spectrometry experiment that's done in the world is defined by these 5 characteristics, and it's how you interpret that information will allow you to uncover new biology. And so what MassQL essentially allows us to do is build the rules and say, okay, this is how we want to filter the data. So for example, if you're interested in iron binding molecules, you can look for iron signatures. If you are doing a drug metabolism study and you know your compound has a halogen on it, let's say chlorine, you can create a query that will filter specifically for chlorinated metabolites, so you can find all the metabolites that contain chlorine. So, but you do need to understand the mass spectrometry a little bit and how to interpret the information. So let's take glycocholic acid, for example. Generally, if you have a trihydroxylated core on a bile acid, what you will see is you see these 2 diagnostic ions. And if you have a modification on there, you see an ion over on the left side. And so we can build rules that basically say, okay, return all scan information basically return all MS2 information, or the fragment spectra, that meet the following conditions, that have those peaks at 337 and 319. Of course, we have some tolerances that we set and some intensity thresholds, but you can filter for it. The other thing that we did is that, let's say, okay, we do see these two ions. Now we have precursor masses. We know we're looking for trihydroxylated bile acids, so we can subtract the mass of this trihydroxylated bile acid to see what the delta mass is. And if we find that, we can report on it. So we built these queries for monohydroxylated, dihydroxylated, trihydroxylated, tetrahydroxylated, and pentahydroxylated bile acids. The most common ones that people know about are dihydroxylated, trihydroxylated, but clearly tetrahydroxylated and pentahydroxylated exist as well. Now, non-hydroxy is not really a bile acid. You need to have a hydroxyl group in this 3 position to be considered a bile acid, but we've included this as well in this particular search. So when we do the searches with those different bile acid mass queries, what we find that we filter 1.2 billion MSMS spectra to about 600,000 MSMS spectra. 21,000 of those are unique spectra, and they represent about 5,500 delta masses. And the way we calculated the delta mass, that if you have a precursor ion minus the unmodified mass, now you could get this delta mass. And so we then took those 21,000 spectra, we searched all of those spectra with MAST and combined them with redo. And now you can start to see the power of connecting all the data in the repository. Because now we can start to see all the bile acids, so color-coded based on the hydroxylation state. The number is the number of modifications. And so what you see is that bile acids in this case are not just distributed into the enterohepatic system, but you find them in distant organs. You can find them in the brain. You can find them in the pancreas. You can find it in the adrenal gland. So, and there are different bile acids that are different in these different locations. We can look at this at rodent data. We can see the same thing happening in humans, where we see these bile acids in the brain. We can see the bile acids in the vaginal region, in the breast milk. And so it's clearly that bile acids, based on this picture alone, is definitely reaching out beyond the enterohepatic system. So the next question we had was, well, can any of these be made by microbes? And this actually used to be a very difficult question to address. But with MicrobeMASST, this is really trivial. We just search all 21,000 spectra. We then report on the delta masses. Some of them we have annotations for, so alanine, GABA, threonine, cysteine, for example. So for those, we have the delta masses on the bottom, and then on the left and the right, we have genera and class of organisms. And what you will see is that organisms such as gamma proteobacteria, Clostridia, bacilli, and actinomycetia are really commonly able to make many of these bile acids, which is pretty remarkable. Now, there's a second thing that we get when we do a mass search. When we do a mass search, we find data sets that have these same metabolites. And if there's some interesting metadata, you may be able to confirm that these are likely going to be made by microbes as well. And so here's a data set that was deposited in 2017, where they had, this was from a cancer immunity paper, but basically where they had mice with a normal community treated with antibiotics. And so what you can see is when they were treated with antibiotics, the majority of these bile acids that we could detect disappeared. So that supports the notion that they're made by microbes. What was really striking is that the 107, this is taurine conjugates. And what we now have is a paper together with Patterson, and it was a back-to-back paper with the Quinn lab, where both labs basically showed that the taurine conjugates are converted by bile salt hydrolase into these new conjugates. And so we now even have the genes that are responsible for this. But this is just one example of the type of enrichment that we can get. We can also begin to link to files and look at the health associations. And so one of the striking things for many of the amino acid conjugates that we saw was that they tended to be associated with IBD. And so now we can go into those public data sets, extract a feature and map their intensity and actually do statistics on it. And what you will find is that many of these tend to be enriched in Crohn's disease patients, but not in relationship to non-IBD controls. And what I find amazing is we get this for free. This is data that's been publicly deposited, where the original depositors did not know about the existence of these metabolites, but we can reinterpret that data, and so we can actually learn new biology. Now, I should note is that we highlight that this is a trihydroxylated, dihydroxylated phenylalanine conjugate, for example. The reason why we are not able to say that this is a cholic acid or a muricholic acid is because the fragmentation patterns are really hard to differentiate. And so at the repository scale search, we cannot yet differentiate, although I do think that is coming soon. But then to provide additional confirmation that this is likely produced by microbes, there's a pediatric IBD cohort, and they had yes and no antibiotics. And what you can see is that some of these new conjugates or amidates are decreased when the infants were provided antibiotics. And again, we get this for free. So, but it's still remining public data, and you want to be able to confirm the information that you get. And so we had synthesized all these bile acids. We sent those synthesized bile acids to the Ramnik Xavier lab, who also is one of the R01 grantees that is part of this knowledge base that's being created. And they had a cohort, an IBD cohort, and they did retention time matching. And the strength of that is that you no longer have to call it a trihydroxylated bile acid, but we can actually call it a cholic acid or deoxycholic acid or chenodeoxycholic acid. But they found 62 of the ones that we sent them. And all the ones with stars are associated with Crohn's disease in relationship to non-IBD controls. What was interesting is that some of these metabolites, such as the methionine conjugates, they tended to be more dominant actually in ulcerative colitis in relationship to even Crohn's disease. And so there's some specificity between Crohn's disease and ulcerative colitis even there. But do they have functions? Of course, that's something we always want to know. So we team up with the Huey lab at UC San Diego. They have a T-cell differentiation assay. And so they looked at IL-17 differentiation, interferon gamma, IL-10, Treg. And so different bile acids tended to activate T cells in different ways, which I think was pretty cool. But because we built this resource, we captured this knowledge, other people use the GNPS ecosystem, as well as all the knowledge that comes into the CMMC knowledge portal. So the community is starting to see these bile acids in their studies, and they're starting to test them against activity. So FXR TGR5 in humans, germination of C. difficile, infants with brain injury that were differentiating some of these bile acids, colorectal cancer, PXR, TGR5 activity, stem cell differentiation activity, intestinal barrier function changes, FXR, hydrocarbon, PPARs, VDR, CAR. So lots of these different activities are beginning to be documented for lots of these bile acids. But what is nice is that all that information in those papers is now in this central resource, so that when you study a specific metabolite, you can recapture that knowledge. So I want to leave you with a summary and some take-home messages. First, we have a community curation system in place. It's ready. We really encourage deposition, curating, and it's really necessary if we want to really mechanistically interpret the microbiome to capture as much of this knowledge as possible in a computer-readable format. And I think that's really important. You know, just because it's in a supporting information in a paper doesn't mean it's readily accessible or findable. Whereas if it's computer readable, and data-driven format, now you can reuse it over and over and over. In the same way that people, when they annotated gene sequences based on functions, biochemical functions that they assign to it by laboratory tests, that is the knowledge that is reusable. And so we're building the same thing, but for microbial metabolites. There is a CMMC website. You're welcome to go to it. There's resources available. All the links are available. The resources are all open and fair compliant. We give hands-on data science workshops. So we have given 3 so far. Both the documentation as well as videos are available through the CMMC website, and we will continue to expand these data science workshops because I think that's really important for for us to learn how people use the ecosystem, but also to make sure that people understand how they can use this particular ecosystem. And finally, I really think it's probably beginning to be time to rewrite textbooks in connection to what we know about microbial bile acids and the roles in biology. Now, I'm in many ways a spokesperson. This is the team that really does all the wonderful work that I described. I already highlighted the names throughout the talk, so you saw their faces with their names. Those were some of the key people that were responsible. I want to thank NIDDK for supporting the Collaborative Microbial Metabolite Center, but also the 100+ scientists that have already contributed to beginning to build this particular ecosystem. And then I would like to acknowledge the Patterson Baker Lab for all the fun interactions we have on bile acids. And then finally, I want to thank you for your attention, and I will take any questions.
Yeah, thank you for the great presentation. Now is our Q&A session. So there's some questions from the chat, so I'm going to repeat those questions first. And then if you have other questions you want to unmute yourself, please raise your hand after these questions. So first question is from Samem. For the gene sequencing of mushrooms, is it necessary to only use data from our own experiment? Or can we also take the sequences from NCBI? Because we usually identify some fungi through microscopy and then match them with references to identify their names, and we don't perform PCR on them anymore, but we can take their sequences from NCBI. Is this also acceptable?
So it's within a limit, it's acceptable. So you know, ideally, if you're going to deposit microbial metabolite information in CMMC, or if we end up including it in MicrobeMASST, we would actually want to know for sure that that's the organism. And so if you know, let's say you're working with Aspergillus terreus, if you know that that happens to be the lovastatin producer, which is a statin used to lower cholesterol. But if that organism, if you're working with that organism and you know it's Aspergillus terreus, and you isolate new metabolites from it, and you learn their activities, yes, you can add that to the ecosystem. We don't require sequencing, we encourage sequencing. In the long term, what sequencing will allow, if you do have sequencing on your particular sample, it will allow 2 things. One, so right now we're in MicrobeMASST, we're only doing taxonomic assignments, but there's a lot of steps between taxonomic assignments. And so let's say you may get a 16S that falls in between taxonomic assignments that are in NCBI. It basically prevents you from doing phylogenetic analysis if you don't have sequencing. And so it's encouraged. But we will capture, we want to capture as much knowledge that you have basically about it. So we'll be willing to do it even if you don't have sequencing, basically.
Okay, and then I think there is a comment from Dr. Maton about during the presentation. Yeah, it's about not.
It was just a comment. Yeah. It wasn't a question. You were saying we get
Well,
this.
it's a fair comment.
Yeah.
The data is not, the data that went into the public repository was not free.
Right.
But it's the data is repurposed in many
Exactly.
ways. But you didn't have to collect the samples. You didn't have to collect the data again. And so in that context, I meant it to be free.
Well, it actually, yes, no, exactly. And and you know, there's always a certain amount of work you have to put into it. But I think your resource, and let me just compliment you on it, I think it is a really spectacularly well done, just shows you why getting all the data and then getting knowledge in a computable form is such a powerful way to approach discovery science. And so that was not meant as a criticism. It was meant as a, yes, you know, this is why it's so important, because now you extract additional value out of data that was collected for an entirely different purpose. And, you know, a lot of people have questions about sharing of long tail data, so to speak, these individual data sets. And they say, what can you learn from them? No one can learn from them. But in fact, we know that you can learn from them. And I think this is a beautiful example of how that occurs.
Thank you. And I didn't take it as criticism. I think it was a fair point. But it's nice that we're getting, at least in the mass spec community and metabolite community, that we're starting to think about reusing data, which hasn't really emerged yet. I think right now, in terms of the metabolomics data that's being shared, it's about 1% at max that gets into the public domain. So there's a lot of data that still needs to be captured.
Okay, so our next question is from Dr. Aziza. I'm curious about the MicrobeMASST. Would it also work to taxonomically search the compound from phytoplankton?
So there are phytoplankton and other organisms in there. This is actually really useful. So the focus of the CMMC is really animal and human microbiome-derived metabolites. But we will take all microbial metabolites. And part of the reason is, for example, Bacillus cereus is generally considered a soil organism. It produces surfactant, but there are also foods such as natto that's consumed in Japan particularly, that actually contains Bacillus cereus. And so you actually occasionally see some of those compounds in there. If you swim in the ocean, or if you eat fish that is exposed to pseudonychia that makes domoic acid, you might end up seeing, and that's a neurotoxin, you might end up seeing that in human samples. And so the more of this data that we have of all microbial metabolites, because you'll start to see general paradigms, because we know certain molecules that are produced, let's say, in an algal invasion of corals, are the same molecules that are produced during exacerbations in cystic fibrosis. And so by capturing as many metabolites as possible, the better chance we're going to have to try to mechanistically microbial metabolites and understanding in animal health and human health. And of course, other people will take this ecosystem and apply it to a very distant application, maybe early drug discovery applications for natural product discovery, or understanding the marine environment. But our focus is really animal and human microbiota. But I think I've now given some examples why we actually want to capture all of the microbial metabolites and their activities.
Okay. So next question is, how will MicrobeMASST include metabolites from environmental sources, as in non-human microbiome sources?
So there's a decent amount. So there's certainly ocean microbes, so Selenospora tropica, that produces selenosporamide that's in stage 3 clinical trials as an anti-cancer agent. It contains cyanobacteria in there. So yeah, we welcome and we would want to see as many organisms as possible. And part of the reason is it actually also helps us to interpret the data. If you want to understand, let's say, human microbiome, and all of a sudden you start to see a metabolite that exists in all microbes across all different ecosystems, then you know that it's not a human-specific molecule. Whereas if you start to see it, let's say, only being produced by Clostridia that are found in humans, then you have higher specificity, and you can say, okay, I will prioritize those particular metabolites for future study because those are dedicated to the human ecosystem. And so by having many other organisms in there, it actually adds a layer of benefit to it. And it helps the interpretation.
Okay, great. So next question from the chat is, how does MicrobeMASST recognize and approve that the metabolites from microbe instead of other organisms? Some metabolites could be produced by plant and microbes, such terpenes. Is that from the mass fragmentation could be known?
So this is a really good question, and it made me realize I wasn't very clear on how MicrobeMASST was really developed. There's a big important component to it, is that all the data that went into MicrobeMASST itself is that the microbes had to be monocultures, so they were cultured on their own without a host. Now we also have data on human cell lines, where we data that were human cell lines that were cultured, and so this is how we're beginning to be able to start asking the question that if the metabolite is found in the host versus microbial. But if we were to do this with complex samples, no, you could not. If you added to the individual metabolites to the CMMC knowledge base, you would need to know that it's a microbial molecule. You may not know what microbe is producing it, because you may have a culture of a microbial community that you're working with. Let's say some of these strategies to continuously grow fecal microbes, for example, as a community. Then you can add just the sequencing link to it. And so then we know that it came from a community.
Okay, so next question is, it seems like MASS only accept a single MS spectral search. Is it possible to query using a batch files with multiple tandem MS spectrum? For example, a bunch of unknown from MS dial?
Yeah, so from MS Dial, so for people who don't know what MS Dial is, it's a wonderful program where you can basically import your data, and it will do the feature extraction. So when I talked about remining the public data for the IBD data sets, we did feature intensity mapping. MS Dial would be one of those programs that you could use to find it, but it also has a sort of an annotation paradigm. Generally, we accept the USI as input. You can manually enter MSMS spectra, so those would be just one each, and you can do as many as you want. There is an API that you can use if you have universal spectrum identifiers, and we take the MGF format that you can search as well. This is how we search the 21,000 in spectra in MicrobeMASST of the bile acids, so that we can see which microbes are able to produce these bile acids. So you do have options of doing at larger scale. I mean, right now, I think we've had 8 million searches already.
Okay, so next question is, many metabolites are not well characterized. Are there any ways to tackle those not functionally annotated metabolites that are derived from the microbial activities?
Oh, that's a really interesting question. So when you have a poorly characterized, so this is one long-term goal of the CMMC, that if we capture enough metabolites and their activities, if you then find that you have related metabolites, you may be able to infer that they may have similar functions. And so that's one way that you can get to function of a microbial metabolite. In some cases, if the microbial metabolites are produced by gene cluster, Selenospora tropica is one of them, they actually have a resistance mechanism almost sometimes on the gene cluster itself. So in that case, they had a copy of a 20S ribosome, and it turns out that selenosporamide is a ribosome inhibitor. And there are many of these kind of examples for small molecules where you actually have the resistance gene next to the producer genes in the organisms that are producing these molecules. So there are sometimes creative ways. But in many ways, if you really want to get to function, you have to create an interesting hypothesis and think about the molecule. What are the functions of related molecules? Maybe that affords you to test it, or do omics technologies to see how they affect biology.
So the next question is, does MicrobeMASST separate metabolites produced specifically for microbe and the metabolites co-produced by host and the microbe?
This is a really great question. So MicrobeMASST does not. So MicrobeMASST is singly based on monocultures of microbes or monocultures of human cell lines. And so there's no co-metabolism in there. We want to actually include that in the future. So if somebody is doing large-scale co-culturing, we would love to hear from you, because we're going to generate the second generation MASS, third generation MASS, where we'll want to include some of these kind of things. Whereas the CMMC knowledge base for the individual metabolites, the knowledge that we try to capture is we do have a host-derived metabolite modified by microbes. It's one of the metadata entries that can be added. So it's a drag-down menu. And that is one of the entries that we had. And I think this is important because a lot of metabolites, they go through the enterohepatic circulation. They may be modified in the gut. They go to the liver. They get modified in the liver and then go back into the gut again. And so there's going to be a lot of co-metabolism that takes place, and we should definitely capture that.
Okay, and there is one, I think it's a comment from the chat, is about the Joint Genome Institute and the EMSL at PNNL. Does metabolic service through competition costs? They would have a lot of data from non-human source, just in case they were not in the sphere.
Yeah, and I would definitely take advantage of this. I mean, PNNL has these amazing resources to collect mass spectrometry data. So if you go through that, make sure you work with them to get fragmentation data, so MSMS signatures, because that's the currency that allows you to do the data science. There's one question that is not addressed yet. It was sent to me directly. So it says, Pieter, nice talk. Just wondered about MAST. If I missed it, if you mentioned it, but is there a way to distinguish hosts versus microbial metabolites, especially if similar molecules are synthesized by both hosts and microbes? Serotonin and GABA, for example. No, because of think of a gene sequence. If the same gene sequence was present in the microbe or the host, you wouldn't be able to, if they're exactly copies of each other, you wouldn't be able to separate those out. But in case of, let's say we found GABA, for example, conjugated to bile acids. We know those are explicitly made by microbes. So sometimes you can make it a linkage not based on that one molecule, but you may have additional downstream molecules that can only be made by microbes, and infer that that pathway is at least active in microbes.